It's not what I focus on, and it's not what I want to put my time and effort into. It is something that I do enjoy talking about occasionally, but yeah, there's a lot of people, like, man, Kane Barlow obviously is one person who puts in a lot of work, and yeah, very grateful to have you here, brother, eh? But yeah, what I want to touch on today is some of the history of alchemy, or sort of some of the primary alchemists in the past, how that's a little like relative to today, and some of the advancements or some of the other discoveries that we've made. So yeah, I'm just going to do my best to glide through this information, and I hope it's well received. I did want to show this slide. Just a little shout out to the male cannabis plants out there. We do see a lot of the beautiful flowers, and although I do a lot of work with those flowers, that's only one half of the genetics and the input. Yep, sure.
Yeah, the boys.
Yeah, the boys. So yeah, I wanted to start it off and just sort of take it all the way back to what is a cell, essentially. I didn't realise just how similar it was. I wanted to find a photo that would show a comparison of mushroom, animal, plant and bacteria cell, but it just literally doesn't exist, because mushroom cells are literally like the same as our cells. They act and behave very similarly. And even I don't know how many people here understand cellular biology, but inside each cell, much like us, we have organs. Cells have organelles, and they do have jobs to do. And yeah, I've obviously noted some of the key differences between these three. A nucleus being the main one between the animal, plant cell and the bacteria. And it does make it a lot easier for that cell to reproduce. Yeah, so actually on my next slide, because I don't remember this all that well, but I wanted to explain some of the basic functions of some of the organelles, and there is reasons for that, because I do want to go a little bit deeper into it and explain some of the ways that the terpene profiles that a lot of us all love and enjoy, you know, what they are and how they're made, and this is how they come about. So obviously the nucleus, I'm sure everybody's familiar with that word. That is the center of the cell. And yeah, this helps, well, a lot of information is actually stored in the nucleus, and when a cell actually separates it, it carries a lot of that information with it. The mitochondria, if we follow the evolution of life essentially, and the theory, I'm studying at uni at the moment, so a lot of my information that I am subjected to revolves around the evolution theory, and it's philosophized or confirmed, or I don't know, something or other in science, that the mitochondria originally when there was just single cell organisms, at some stage along the way, whether it was by divine intervention or just evolution itself, somewhere along the lines a bacteria and a single cell basically decided to get along, and instead of the cell eating the bacteria decided to create a new function inside the cell and produce the energy to fuel literally everything else that sort of happens in the cell. Your ribosomes is where your proteins are translated and created. Well, so not so much. The endoplasmic reticulum is where your proteins are mainly synthesized, and the Golgi apparatus, packages your proteins and lipids and sends them off to where they need to go. The only reason why I sort of wanted to just articulate on this is to just open up people's minds. If you've never seen this literally blew my mind first seeing this under a microscope, and then as I've delved deeper into understanding medicines and just life in itself, it's been really helpful to understand how adaptations and evolution and how we can change things and work with things in order to see different results. This is something that I actually did in first year science in biology at uni, and it's basically when you look at well, in this case, in this example, I've basically frozen these cells in time. What you can actually see here is various stages of meiosis, which is cell duplication. So at these different stages, when a cell first starts to separate, it starts to wind up all the DNA, and it, everybody's heard of what a chromosome is, yeah? Well, what's actually happening when this cell is winding up all of the DNA and slowly stretching out to become another cell is it's actually turning into chromosomes, and those chromosomes, what they are is basically regions of genetic information, and when this cell stretches out to create both like two new cells, it needs all that information to replicate and to create the other cell. So that's essentially stages of that you can see at the moment. And I've just gone into a little bit more detail because I do want to talk about something that's pretty cool, and I'm not sure if it's a safe topic to talk about, but I do want to talk about it. It's got nothing to do with gender, holy dooly. So these are your five main stages of mitosis. Sorry, I was incorrect calling it meiosis before, but that's a different thing. So basically, what I do want to speak about some important things, so I can speak about next is at the point of the prophase where the spindle fibers emerge from the centrosomes. They basically tell the cell to start raveling up your genes, your DNA, and start raveling it into chromosomes, essentially. And another point that I want to make, which it may not actually be on here, no, nothing about the telomere ends, that's alright. So, yeah, basically just wanting to highlight the differences in the stages, and I do want to speak about something, like I said, it is a little bit controversial, but I'm studying genetics at uni at the moment, so I have done various processes such as transgenic insertions of genes into other plants, like I made Nicotiana Bethlehemia glow in the dark by inserting a gene from a bioluminescent something or other. I hope my lecturer's not watching this because I was supposed to know what that actually came from, but it came from something, right?
It came from jellyfish.
Yeah? I'm just gonna, yeah? You're familiar with that, are you, Bella?
I'm a geneticist.
That's beautiful. Thank you very much, bro. I appreciate that. There you go, ladies and gentlemen. But yeah, so something that I do actually find a little bit interesting, that, like I said, it is a little bit controversial, but this, to my knowledge, and you may be able to speak on this a bit more, hasn't ever been seen in nature to this degree, and I'm open to be completely criticised by that, brother. But the really interesting thing in chromosome 2 is that what we're actually witnessing is a fusion between the two chromosomes, and it's so recent that we can actually see that it actually has two centromeres, only one of them is active. This does happen in nature, evolution, chromosomes do join, and that is a natural selection thing, or I guess you would call it a mutation. Would you call that a mutation, brother? When chromosomes fuse, or just a fusion?
Yeah, I guess it would be a fusion.
Yeah.
Yeah, but it's a tough thing.
Yeah. Usually when this happens, it's very messy, and you can see this in the genetics, and you can also see it with the way that essentially a new genetic is trying to cross with an original genetic, and generally it wouldn't be something that would be accepted in a genetic crossover, essentially. So some of the really interesting things that make this stand out from others is that it has telomere sequences in the middle. Normally these are found at chromosome ends, but they appear in the center of chromosome 2. And as I mentioned before, the dual centromeres, only one of them's active, so one of them has turned off, and that's obviously what it allows. What allows that to have a correct function in there. I really want to talk about this because I do find it interesting. Whether it was a a natural evolutionary phase of our existence, or whether it was some kind of divine intervention, or maybe people who have been doing science longer than we know. I really don't know, but I just thought I might add this in this discussion because I actually find it really interesting. So, because this is the Mardi Gras, I thought I would try and explain the roles of evolution, and in turn, like the way that new terpenes are produced, and some of the reasons why that we know of. When cannabis undergoes different selection pressures, as you can see, from pests and pathogens, different sunlight, nutrient profiles, water, and pollinators. These are, obviously this is very simple things to think about, but in nature they can become very complex. If there are any growers here, you'll probably be familiar with the language of naturalization. It takes about 30 years for a cannabis plant to naturalize. During this time, it might need a little bit of help. It might not be the best looking plant, but if it does get to this point where it's actually reproducing its seeds without any help and growing on its own, that is the naturalization process. So it takes time for enzymes to evolve and mutate. Obviously, selection pressures favour certain terpene profiles. Over hundreds of years of enzyme changes get locked into DNA through natural selection. So when you look at that cell from the start of the image, each one of those cells have a job, and slight changes in food, environment, literally anything, any change in anything that's not a constant is going to bring about modification in genetics. Whether those modifications are permanent or whether they're just seen momentarily is another case. It really depends on whether what you choose to do with your plant or what nature has in store for it, essentially. The thing that excites me about this is that this does mean new medicines, any new pathways in cellular biology that produces new compounds. It does mean just in turn that we have new things that we can study and that need to be looked at. And the really cool thing about the cannabis plant is that it has genes for hundreds of minor cannabinoids, terpenes that can be upregulated or lost depending on environment. And this is what's really fun about the hunt for land races. I don't know if anybody is a grower or is interested in genetics and so on and so forth, and finding new medicines. The cannabis plant is so interesting because it really does just have so many terpenes, and just from all other places around the world and other living organisms and plants that produce the same or similar profiles. They're all in this plant, and that's pretty crazy in itself. So yeah, so obviously for me personally, the understanding of this inspires me to obviously learn more, and I am really interested in medicinal properties and finding new medicines, and just being a part of the journey of research, and yeah, just really intrigued or invested to be a part of this. And this has led me down the journey of understanding how to, well, I first started doing various extractions. When I feel like I started understanding medicine a little bit better, I started getting more into using tinctures and dual extracts, so I can get as much of that whole plant medicine or whole mushroom medicine as I can. And trying to understand a little bit of the history of some of the things that led to these practices being common knowledge is pretty cool to take a trip down memory lane. And I have just outlined some of the interesting moments in time. I can't actually see it too well, but as you can see, from 3000 BC, the Egyptians were using wine and vinegar to extract, to use various plants. Mesopotamians, 2000 BC, using alcohol-based extracts also. And you can see this common theme. Ayurveda are people familiar with Ayurveda and what this historical record of medicine is? Some yeses and a no, sir. Ayurveda is, to my knowledge, the oldest written context of medical practices that we still have. Unfortunately, a lot of that information was destroyed, but this is the oldest source of information on these particular types of healing properties of plants that we have. Yeah, and as you can see coming through, I just wanted to show some instances where these practices have been used, and as time has gone on, things have obviously become more complex, and there have been more systematic approaches to the ways that we can perform extractions and the bio sciences behind it. Does anybody know who Paracelsus is?
I know he's dead.
Yeah. Does anybody know what spagyrics is? Yeah. So Paracelsus was the he's kind of coined as the father of. He's kind of coined as the father of alchemical medicine. Some of the notable things that I think that should be mentioned is his journey with not just at the time there was a lot of all your alchemy was based on you know, trying to make gold or trying to turn minerals into more valuable minerals, and Paracelsus was the first person who, to my knowledge and to what I can find in texts, was the first person that sort of wanted to start tinkering with that and using it and finding out what it can do inside of him. And he didn't pioneer these understandings in regards to transmutation and alchemy, but the idea, even back then, without understanding these cellular functions and without having a microscope, he did understand that there was things happening inside of us and inside of other living organisms that will change one compound into another compound, or various compounds into other various compounds. And he labelled this transmutation. If I could break it down, it's sort of like transmutation happens inside, and alchemy is an external form of that. If you want to play in those fields, and I think I've just noted that as well. So I just thought I'd summarise this for myself, even for my palm cards. But like I mentioned, unlike other alchemists focusing on transmuting base metals into gold, he was really super focused on revolutionising plants and finding what properties that we can use. He viewed alchemy as a tool to unlock nature's healing potential, and obviously I already mentioned transmutation and alchemy being foundations of his understanding, essentially. I find it interesting, and I just thought to mention as well, there is a bit of a theory within spagyrics at the time, and between the two solvent extracts, you would have your in an ethanol extract, you would generally have your terpenes and your various other compounds. The polarity will extract all of those, and when you want to do a dual extraction, you use water as another solvent, and that can extract your polysaccharides, and we call that a dual extract. However, back in the days with spagyrics, if you could imagine a cell and all of these beautiful medicines and compounds inside of that cell's designed to protect itself from all kinds of things, really. So modern extraction methods focus on trying to get through that cellular membrane and extracting those compounds out. But the spagyrics method is a little bit different, and it's really interesting because what they would do is they would use a yeast and sugar, and depending on the plant, obviously, because there's all different sugar contents, practice makes perfect, I suppose. They would, instead of working on ways to disrupt the cellular membrane, they'd literally just eat it with the yeast, and that would actually release all of the chemicals inside of it. But one of the really interesting things as well is that if you take like a mushroom cell, for example, the outside of it's made up of chitin, which is really, really hard for our body to break down. But fortunately, things like Saccharomyces genus, or Saccharomyces cerevisiae, produces various enzymes. This is how mushrooms break down their food. This is how, well, I guess we all break down our food actually, us animals and mushrooms. And this is just an example of some of the enzymes and some of their functions and what that actually means. Some of these compounds that they are breaking down is really important. We don't really want those in our medicine, and having more bioavailable material is a lot more helpful in the long run. Yeah, I wanted to introduce things with a bit of a background on cellular functions, and just explaining some of the basic alchemy practices from the past. And with that in mind, yeah, I wanted to show you this slide. If you've seen mushroom talks in the past, you've probably seen stuff like this, but I think it's really important for the people that haven't to understand what a mushroom is. Most people associate what we can see as the mushroom in itself, but that's only that's the sexual reproductive system of the mushroom. Most of its time is actually spent, as you can see, as these different mycelium structures. This is showing an image of a mushroom that you can obviously see it's growing out of the lawn essentially, or the ground. Mushrooms inhabit all spaces, and yeah, they have their favourite foods, whether it's an acacia tree or a big ironbark or whatever the case be. But I think it's really important to look at this, and you can understand a little bit better about how these mushrooms actually function and what they're actually doing. So yeah, I just wanted to basically make a point of explaining the differences in some of these types of mushrooms and their different ecological roles. Not all mushrooms get along, and obviously some do. The same with plants. Yeah, some plants just aren't going to like each other and actually try to kill each other. And that can be seen throughout all forms of nature. When people talk about mycorrhizal mushrooms and the trees being able to communicate, there are mushrooms that can do that and do help. But I do want to make a point of letting everybody know that although a mushroom, from your perspective, looks beautiful and peaceful, there's probably some pretty gangster shit going on underneath it for it to get there. So as you can see, I've just highlighted 4 types: decomposers, pathogens, symbionts, and endophytes. And furthermore, I wanted to reinstate this in this particular way. Trophic modes. The significance of this is, what I want to show is the difference between these 3. So a saprophytic mushroom, we have saprophytes and primary saprophytes. Saprophyte mushrooms, as soon as a tree dies, they're your first mushrooms that activate and start eating that tree. Your primary saprophytes, obviously the name denotes that they are first in, best dressed sort of style. I will share this first, because it's interesting to share it in this moment, I think. I just thought. Has anybody heard of brown rot or white rot fungus? Yeah, some people have. Cool. The reason why coal even exists is because previously, before a particular evolutionary trait that allowed mushrooms to produce an enzyme that eats lignin. We had a world of different mushrooms essentially, and these mushrooms, some of them are known as what's called brown rot fungus. So these mushrooms would eat the trees, but they would actually leave the lignin behind, and throughout, I don't know how much time, let's say a lot more than the birthdays I'm going to have, those trees actually become coal. At some point in our history, mushrooms developed the ability to eat lignin, and that's why no coal is being produced anymore. What's really interesting is that the species of mushrooms that fall into the saprophytes and those white rot fungus are literally a lot of the mushrooms that you guys probably know, or what I know as well. A lot of our medicinal and edible mushrooms, psilocybin mushrooms, fall into that category. Lion's mane, reishi, pretty much, yeah, most of them. So I do find that a little bit interesting. It is a later addition, evolution-wise, to mushrooms. Yeah, everybody's probably seen cordyceps photos before as well, yeah? They're not the only parasitic mushrooms. Some trees fall victim to particular parasitic mushrooms. Some mushrooms can actually start off as parasitic and then turn into saprophytic when the plant dies. So this is something that they've learned to do. And yeah, it's not much fun for the plant, obviously. And that's the plant's job, to produce, to evolve, and to produce new metabolites and new enzymes to help them protect themselves from these mushrooms as well. And obviously our mutualistic mushrooms, there's all kinds of mushrooms that we can use to increase the nutrients, increase bio availability, and just increase, just have it's more ecological, sustainable unity between organisms, just creates a more healthier habitat. I got asked to speak on cubensis mushrooms at my last talk, and I thought it would be a really interesting thing to add into this presentation. Some of the things that we can see over time through just selective breeding that us as people have been able to view in real time. All dogs just about used to be wolves. If that's not an indication into what we can actually achieve with a particular species, then yeah, it's another good one is magic mushrooms and cannabis. I think between cannabis and magic mushrooms, for any plant grower and any mushroom grower, whether you want to grow those particular plants or mushrooms, because of the industries behind these particular movements, there's so many people that have just been like cowboy genetics, crossing this with that, trying new things. It may not be the brightest idea. Or I know 3 years ago I messaged Kane Barlow with an idea, and he wasn't rude to me, but he just told me, he's like, that doesn't make any sense, mate. Like, what do you want to try that for? And it was, he's a humble man, but I was just sort of like, oh, damn it, that's not going to work. And then you know, as time goes on, I'm like, that was a dumb idea. I better not message him anything for a little while, eh? Because I don't want to feel like that again. But in the psilocybin growing industry, I think they call it something like ghetto crossing. There's complex ways that we can breed mushrooms, and that's all really interesting. A really easy way is to just flick some spores onto a petri dish and just see how they go and look at the end product. And much like when you're looking for a phenotype in a cannabis strain or whatever the case be, you can visually see the difference. And this is science. That is the scientific method, you know. But yeah, I sort of just wanted to show some variations in these cubensis strains. I think it's Darwin sort of, not his exact words, but sort of mentions that anytime you domesticate any species, you will see mutations, you will see problems, and whether those problems are desired or not is up for debate if this is your goal. But as you can see here, this here is just probably what a lot of us, if you have been out picking gold tops, is probably how you will see them. You may see some nice fresh ones like this. I just wanted to give this little bit of an indication because something really close to a wild mushroom cultivated would be something like this. As you can see, there are some differences. To growers, you know, could probably list some really serious descriptions, but to the average person, maybe not so much. It doesn't take long when you're growing mushrooms, when you're giving them what you think, or when you're following a tech, whatever you're giving them. It doesn't take many generations for them to adapt and for them to change. And what you can actually see here is, I don't know what people's experiences are with strains of mushrooms or their understanding, but quite a famous cubensis that came out some time ago is actually known as albino penis envy. And this is this one here, obviously, or you know, somewhat obviously. What you can actually see here is a mutation that has caused albinoism. Two really interesting mutations that we can see in mushrooms is leucism and albinoism. The real difference is one's spores are blue and one's spores are black, but obviously there are genetic differences. And ironically, although these mushrooms look really fucked up compared to what they're supposed to be, they produce massive amounts of psilocybin, and for people that are growing magic mushrooms in general, that seems to be the goal. So that's mission accomplished, obviously. But as you can see, things have changed a little bit. I think this is Jack Frost, a strain that people may or may not be familiar with, and that is a love child of this particular mushroom. And Enigma, is that from ape as well? Yeah, so that's another genetic mutation that has come from that direct lineage as well. I just wanted to include some of these photos because that Enigma doesn't look anything like the mushroom that you can see in the center. And I guess it's up to whoever wants to do what, you can do whatever you want, eh? But as you can see, if you do tamper with stuff, it's going to produce some really seriously, I would argue that some of those mushrooms are sick, and they're never going to survive outside of whatever environment that it is that you're growing on. Something that I have focused on for most of my life, and as I am starting to learn and delve deeper into mushrooms, something that I've carried over as well. I'm first generation Australian, my family's from Ireland. I really respect this land. I'm obviously not, my ancestors don't come from here, but I have a very deep connection with my own land and a very real history. And I feel very passionate about making sure that things of this land are sustained and kept. A lot of these things are very valuable, and maybe I don't even fully understand the meaning of that, but I think I'm learning more and more as the days go by just how important that is. For about 10 years now, I've been going on walks with a group called the Queensland Mycological Society, and it's a group that, it's a great group, honestly, a great group of people. It's all mushroom people, yeah. When we were going for a walk, it was my first walk with these guys somewhere in Brisbane, I can't remember where it was. They shared a story about how, you know, some years ago they were going to get rid of this parkland, and how they actually managed to stop that from happening was by proving that there was a mushroom that can only be found there. It was 10 years ago, it was new, I was new in my journey, but it stuck with me. I don't remember the details to this story, but it's always stuck with me to just to keep that in mind. When growing any plant, if you're growing a native plant to this land, it's generally, if it's from that space, it's illegal to take it out as well. On my journey of trying to find compounds in native species, I thought it was really important to utilise this idea, because for me, I love tinkering with medicines, alchemy, finding new things. Every single thing is in the bush all around us right now. Every single thing, you know, maybe not some things, but a lot, like everything that most of us could want or need is all around us. It's just about establishing better relationships and understanding these other living organisms that have grown with us. I found a really interesting photo from Paracelsus's days. I can't remember what it was. I was trying to Google it. It's in a book. I wanted to share it with everybody. But it's a photo of an old alchemist, and he's holding a lantern. It's at night, and he's leading the plants to a destination at the same time while they're leading him, following the moon, and he's actually following a woman. So he's actually following a woman. And I just found it, I found it really interesting that that understanding was, was all the way back then. And yeah, I like to think of things a little bit in that way, in regards to our relationship with these other living organisms and the way we can interact with them and the way that we've evolved together, essentially. Obviously, securing food and medicine, that's a big deal, and protecting our ancestors. These, on a very serious note, as real as it gets, we've all, it doesn't matter what people's beliefs are, but for us all to be sitting here right now, to even just eat a hamburger, to even smoke a bit of weed, you know, like a lot of things have gone into play for us to just, to just be. So I guess that's something that ripples with me, and I try to keep with me as well and remember. On my mushroom journey, and always trying to find new things and learn about new medicines. I've been working with these particular mushrooms for quite some time. I personally use Usnea every single day. I had a really long battle with various bacterial infections, and going to various doctors and then prescribing antibiotics and not wanting to prescribe antibiotics at times, and you know, the list goes on with the trials and tribulations that I felt like I was experiencing. And I ended up just thinking, fuck it, like, I thought I can just make my own antibiotics, you know? And as I'm studying at uni at the moment, I get the opportunity to do various projects and assignments. Yeah, and pretty much up to date, I haven't really had to worry about choosing anything for my assignments, because I can just choose my mushrooms and heavily research them, which is fun. But some of the things that, well, these two mushrooms in particular, Trametes coccinea. Has anybody seen that orange mushroom? It's pretty common, yeah? So a really interesting thing about this, a professor from QUT told me in 2020, this has what's called cinnamomanic acid, and also another unknown antibiotic. Historically, in this country, there is one article that speaks on this mushroom with indigenous use, and it's mentioned that it was used as a teething thing for children, mouth ulcers. Can't remember from memory, but a few little things, and it's not a big mention, but as I started to research this mushroom, and in particular cinnamomanic acid, I found out some really interesting things about this one particular compound. So the kynurenine pathway metabolite, the kynurenine pathway is a really important conversation in neuroscience, and this particular metabolite is the single most understudied metabolite in that pathway. There's only 40 papers in the world on this particular compound. I just found that really interesting, when this actually has significant impacts on psychological ailments, in particular schizophrenia and psychotic episodes. And if anybody is interested, I would encourage to look into it, as I am at the moment, because I do find that very interesting. This antibiotic just kills strep throat, just completely destroys it. There's it's a really powerful antibiotic, and yeah, I think it should. I think a lot more research would do a lot of good. I can't remember which one, but one of these antibiotics kills all gram-negative bacteria, and the other one kills all gram-positive bacteria. So it's a pretty interesting new old antibiotic that we can work with, and yeah.
Maybe can we just cook and chew those? Cook and taste and chew.
Cook until like so make a tea sort of thing.
Yeah, or just cook it up and then taste it and then chew it.
Yeah, I'm just thinking about the boiling points of all the different compounds in there. I know that
I was just thinking what the Aboriginals were doing, because they were giving it, were they just eating it raw, or did they heat it up?
Yeah, so to my knowledge, that's the extent of it, is yeah, just chewing on it. There's literally no more information besides one article on that. I couldn't answer that, truth be told.
It was almost just dried or raw.
Dry or raw?
Dry or raw, yeah. Yeah.
Maybe a make poultice out of it, maybe. Make poultice.
Yeah.
They're all good ideas, you know. Like I was even looking into eucinic acid because a really interesting thing about it is that it has what was I had it. So that's right. So both of these antibiotics, one of their cellular mechanisms. If you ever want to look up how something works, some kind of drug or some kind of medicine, look up cellular mechanisms, and you'll start to like see what's going on and what they're actually doing. And one of the really interesting cellular mechanisms about both of them is they actually destroy the biofilm around bacteria. So biofilm formation is a really big problem in bacterial resistance in medicine. Now this handles it pretty well, and the idea of like using it in more resistant strains of bacteria or cases where maybe like spraying with ethanol in a hospital environment, you know, spraying with ethanol, it might not fully kill that bacteria because it's protected in a biofilm. I've thought about even just the idea of even adding a little bit of that to a spray, you know, just to increase that effectiveness of whatever it is that you're using. Because you can use various things together, but you do have to be careful if you are going to be combining things. It's not really advised unless you really do know what you're doing. Some people give advice about combining certain medicines, and it's not generally the best idea. You can't just put 10 mushrooms that say that they're medicinal and whack them all together and say like, this is great. Although chances are it could be great, but there could be things that compete with each other, and there could be some things that just disallow other compounds to be absorbed through competitive inefficient. Yeah, I think, yeah, that's all I've got for you today, guys. Thank you.
Right, we've got time for some questions. Thanks, Liam, for a really interesting talk. I'd just like to remind everyone that this is a collaboration between Mardi Gras and EGA. So if you're liking the things on in 10, we've got some flyers up here and there's a bit more information on where to find them. So, has anyone got any questions for Liam? Cool. I don't know, bring the mic over.
So I missed the first part of your talk.
Start again?
Well, maybe, I mean, it's been recorded. So I do natural land regeneration around this region, and I've heard anecdotally that only 30% of Australian mushroom species have actually been identified. Do you have any input, or have you received any information regarding the validity of that?
Well,
what would your take be?
I know I have seen a presentation on that topic in particular before, but as far as what percentage that is, what we do know, it's very, very small. It would be 3 or 3, like 10%, but 10%, but there you go. We got Kane knows.
10%?
Yeah, and that's the really exciting thing, because that's only 10% of the total.
Knowing variety.
Yeah, and then there's just so much to discover. When a friend of mine found a Psilocybe subaeruginosa, and he spore printed it, and he was growing it on a plate, and he mentioned that it looked so aggressive and so strong, very ropey, very stringy, and the idea that this would surely this looks strong, like this must break down something serious. And he contacted a professor at the uni, and it was really simple. The professor just said to get some organic red wine, water it down to 25%, make a liquid culture, sterilise it, add a little bit to your culture, your liquid culture, let it adjust to that food, and then chuck a nice dollop in your new liquid culture, red wine. And you know, some mushrooms will clear it up a little bit. This particular mushroom, it turned it into clear water, you know, it ate everything. So what that actually means is that the 300 and something or 400 and something naughty compounds in red wine, that basic building blocks for some of the nastiest compounds that we actually know, these phenols, this mushroom eats all that shit, and it's a matter of finding out exactly what we can use that mushroom for, all kinds of mushrooms for, in these regenerative opportunities, you know, to clean up some of our messes and just work closer with the land, eh?
So it's wild to think about how many functions and chemicals we just don't know about, or are actually wiping out the habitat of.
Impossible to think about, eh? Yeah. To think about that throughout history is just, it's amazing. What's your name on Instagram?
I don't really use Instagram.
You're not from around here? Did you say you're using...
I'm local, I'm born and bred.
Yep.
I do land care around the region.
Okay, yeah.
So we're often coming across a lot of mushrooms that you just never see photos of. You go look through mushroom identification books and you just, not in here. Then aside from that, I'm way into biochemistry and neurochemistry, so it's sort of trying to associate different things, like what you're saying about, you know, what was it? Talk a bit closer. About different, compounds that we, what was the question about, someone asked a question about how certain things were used, and we didn't understand necessarily how it's used and the potential benefits. Yeah. So like, how much of that is actually lost? Like, they call it the stolen generation and the lost knowledge. So like, I know several different native plants to around here that have side uses, from little insignificant things like eating, I mean that's not that insignificant, but like washing your hands, like the soap tree, to things that were used as poultices or say anti-pregnancy, like kangaroo apple for example.
That was used anti-pregnancy, yeah.
Yeah, so like a lot of that knowledge isn't actually being retained to the next generation, and then at the same time, I mean to modern colonists in our modern society, I should say. So like, we're in turn losing so much of that, not just from losing the cultural companionship, I guess, with Indigenous Australians, but also through land destruction and habitat loss, species loss, and a lot of that we don't even know what species, like with mushrooms, if we only know about 10% of mushrooms, and it's like how many species are we losing before we've even seen?
Yeah.
And I find that quite fascinating.
It is. It's fascinating, and if you get sad sometimes, and you know, and feel a bit sentimental too, eh, brother? You know, that's history, man. Like, you pick apart of the world, colonization, and the way that it's enforced on people. You know, part of taking people away from themselves is taking them from their land, taking them from their spirituality, taking them from their knowledge, all of that. So it's no secret that that is what's been going on in many places, and unfortunately we don't have that kind of information, you know. If you look at things like Ayurveda, or when Alexandria come and destroyed all the libraries and that, yeah, all the books, You know, that's all that we do have left. I can't remember how this quote goes, but someone said it to me and it sounded real fancy. But it was something about like in regards to science, you can't if you take away all the science, the truth will come back because the science is still there. You just have to rediscover it or discover it. But you can't do that with lies. Yeah. So.
Yeah, that's a big sentiment in Ayurveda.
Is it? Yeah.
Thank you very much.
No, well, thank you, brother. Yeah, thank you.
Right, has anyone else got any questions for Liam?
I just want to add an extra point to that. I think part of this journey is also, you know, like learning how to grow these fungi within a lab environment, but then with the potential to then potentially reintroduce them back into the you know, their ecology essentially. Like,
Yeah.
you know, like
You're right.
we find these kind of new mushrooms, we find these species, and I feel like, I don't know, maybe it's just maybe it's a human thing, maybe, I don't know, but I certainly see, given the damage that we're doing, that there is some responsibility in learning how to work with these fungi and learning how to reintroduce them.
Yeah, definitely, bro. And I think
Appreciate the work.
well, thank you, and you already know how I feel about you, brother. Thank you.
And like a lot of these domesticated species, these domesticated strains, they won't survive. You put them outside, they're gone.
Yeah,
They're incredibly weak.
Yeah, for most of them, yeah, we do have other invasive mushrooms, but
Yeah, they're not, but they're not domesticated in the lab, like, you know.
so no domesticated lion's mane, domesticated reishi could escape,
Like, I mean,
even
in
if it's just
theory,
one
yeah.
generation.
Yeah,
Like,
in theory, yes, but then you wouldn't call it domesticated because it's not like it's, you know, when you make penis envy, it's like there's a significant visual and chemical element, like you can measure or see that's different.
yeah.
Like there's a mutation, like albinism or something.
How many generations do you think? Because it's three generations for cubes, yeah, to see significant genetic change in cultivation. No.
I think we're all in the line of 10 generations.
Okay. Yep.
But see, a lot of these mutations, like penis envy, there's something that randomly turns up
Yeah.
and starts in their core, and then they're propagated onwards from there. And all the other strange receptors got in that process.
That's right. Yeah.
So
Yeah.
it's like that's the desirable characteristic for them.
Yeah.
So we're going to continue that line
Yeah.
from there onwards.
Yeah. And does obviously that would create recessive genes as well, just crossing them with their own spores as well, yeah?
Or they're inbred at that point.
Yeah,
And that is the inbreeding that causes part of that mutation.
yeah, yeah,
So,
yeah.
Or even the selection of things that are already there that just would never survive, but you select it and
Yeah.
take it, and then it's like, it wouldn't survive anyway, so but you just make it survive.
Yeah, yeah. Sweet. Well, thank you very much, everybody. If
I
that's
think
all-
we've got more questions.
Sorry, brother.
Thank you. Speaking to the land care chat, you can get involved with citizens science through the herbarium by adding to it, because like you said, there's very little knowledge on it. And on that point to you, sir, who's leading the who's leading the way with analysing subtropical and let's just say native mushrooms in Australia for their alkaloids and compounds?
Pick a genus, brother. There's people and I think this is the really beautiful thing about it. Like, I know I've got my little favourite little genuses I'm trying to get to know more, and I do know of different scientists and people who sort of hyper focused on certain things that I admire. That I'm like, oh well, I'm never going to get that deep with my work on Agaricus genus, but I know, this woman is doing a lot of work. She's, or whatever the case be, bro. But to truly answer your question, I don't know who one is, but there's many groups, bro, and there's such great people in the industry with so much knowledge. To put a finger on that, sorry, yeah.
is there like a collective database that people are all adding in their information from around the country and around the world on Australian native fungi?
There's a few little groups, but as in regards to like actual like
Open
terpene alkaloid profiles, what I've been doing, brother, my truth right now, I've been collecting every single medicinal mushroom that you know of, bar chaga and cordyceps. I have a native species, bro. What I've actually been doing is looking at the lion's mane, you know, the compounds in that, going, well, hold up, it's over here too. Well, different levels, you know, much like a plant, different levels of alkaloids in certain parts of it too, you know, the mycelium, the fruiting body, and so on and so forth.
Different areas, different geographical areas as well.
Yeah, well, if you take like a reishi, for example, or the genus Ganoderma, you know, the multiple species that we just have in this country with a different profile of compounds that we can utilise, and they all have their own synergistic effects, you know. If they wouldn't be a species if there wasn't a reason why they could produce those sorts of things, so I think that's my time up, is it?
It is.
Yeah.
Can you all join me in thanking Liam for his interesting talk?
Thank you.